Okay, so we are going to now start on glycogen. We will look at two things. One is going to be breakdown, then synthesis. No, that is glucagon. So don't confuse glucagon and glycogen. There are two different things, totally different things. Glucagon is the hormone, glycogen is polysaccharide. It's a polysaccharide of what? Glucose. And is it a branched polysaccharide or linear polysaccharide? Branched. So it has, what type of linkages does it have? 1, 4 and 1, 6. So that's glycogen. So it has alpha 1, 4, and so this is glucose, and then we got alpha 1, 6 glucose. So this is, which is the linear part, which is the branched part?
1 to 4 is linear and 1 to 6.
And about how often do you see branching in glycogen? About 10 to 12 residues, right? So 10 to 12. And in this case, by residue, I mean glucose monomer, right? That's what I mean in this context. Okay, so why there are so many branching points then in glycogen? Are there more branching points in glycogen or in starch? Glycogen is more branched, right? Why is that? Right, so more linkages will give you more branching point, but why would glycogen have more branching points than starch? So we have more branching points in glycogen. That means that the glucose can be easily accessed, right? More branching points mean easy access to glucose when we need it. We quickly need glycogen. Plants don't walk or anything, right? We do walking and all the other activities much more than plants do. So this is a mechanism by which we get quick release of glucose from glycogen breakdown. What do you remember about the structure of glycogen? Huh? It looks like a tree. Okay. And then what about how many reducing and how many non-reducing ends are there? One reducing end. And how many non-reducing ends? A lot, right? A lot of non-reducing ends. So when glucose is released from glycogen, from which end do you think glucose is released? Reducing end or non-reducing end? Non-reducing end. There's only one reducing end, so it don't make sense to release glucose from the reducing end. So it is released from the non-reducing end. So the mechanisms are in place to release glucose from the non-reducing end. So if you have a 70 kg weight, body weight, approximately 100 gram will be in the liver, and about 400 grams will be in the muscle, because there's a lot more muscle weight than liver weight, right? So it makes sense to have more weight of glycogen in the muscle than in the liver. How long do you think glycogen will last when you are starving condition? About 8 to 12 hours, right? So it lasts about 8 to 12 hours when fasting. We get rapid access to glucose. So the structure of glycogen looks something like this. In the center, there's a protein, and this protein is called glycogenin. We'll see in a minute. And this protein is a dimer. So always it will have a primer linked to it. It turns out the enzyme that makes glycogen will not work without a primer. Primer is about 8 glucose units. Okay, so 8 glucose units, and they are always attached to this protein. So this is the primer, and then we'll have secondary points, branching points. And so let's say like that. So that's your, this is your first tier, that's your second tier. And then we'll have more. So this is how it is going to propagate all over the core. So that's the core, and glycogen will propagate like that. And so each of these are about 12 units, right? Each of these branching points are about 12 units. So you're adding glucose units at the non-reducing end, and this is how you extend the chain. So approximately there will be about 55,000 glucose units, and molecular weight for these are very high, more than 1 million. One reducing end, lot non-reducing end. Oh, this is reducing, sorry. All of these are non-reducing ends, yes. Only one reducing end, which is always bound to the protein. One reducing end will be attached to the protein at all times. And this is the starting point to make the branches on this side. That's the starting point to make the branches on the other side. Once you have it in the liver, once you see it under microscope, you'll only see as granules, right? So that's the whole glycogen, and because it stays like that, this will be your glycogen. There will be about 12 tiers of glucose units, or the chains. So about 12 chains. Let's make glycogen. So glycogen synthase needs two enzymes. One is glycogenine. So glycogenine is the protein that produces, so this will produce, and so this will be your primer. Before I go, there's one important thing we have to really get across at this point, which is, in general, when you have sugar that is being utilized to polymerize, we will use G1P, not G6P. So what we have seen so far is the glucose went to G6P, right? Then we did other things. But whenever we are doing glucose as a polymer, storing it, we have to use G1P. So what will happen is that G6P has to go to G1P. So which enzyme does it? Could do it. Could you predict an enzyme? Mutase. So this is a phosphoglucomutase. Very important. The second point I'm going to make is that in addition to G1P as the trigger for storage, sugars will be associated with the nucleotide, which is also as the signal for storage. So UTP is going to be our friend when we are making polymer of glucose. So UTP would look like that. You'll have uracil here, and then that's your sugar. So this is our UTP, uridine triphosphate, where you have the base, that's your sugar, and that's the triphosphate. That's our G1P. How are we going to make a bond? So which is a better nucleophile, you think? Or rather, which part is a better leaving group? UTP. Why is that? Charges, and it has it has three phosphates, right? So this is a good leaving group. So we're going to make UDP glucose. How many phosphates are there right now? 4. So if you make a diphosphate, 2 has to be released. How would that be released? This will attack here, and then we're going to release this, right? So that will be lost as PPI. So once you do that, then this is what you're going to make. So this is our UDP glucose, and in this process we released PPI. This PPI will be hydrolyzed to PI. Do you expect this to be hexagonic or endagonic? Hexagonic. Hydrolysis reactions are always hexagonic. So this will have a highly negative delta G, and that is what is driving this process to make UDP glucose. So first thing to remember, whenever you see glucose as a storage polymer, then G1P, but G1P alone is not going to work. We need to have UDP glucose, and again attaching it to a nucleotide, the trigger or the signal in the cell is that that pool has been assigned for storage. The cells keep separate pools from G6P, right? G6P is totally separate. That one is not a storage signal. G1P is a storage signal, but G1P alone is not going to do it. So it is associated with the nucleotide, in this case UDP glucose, and when you see UDP glucose, the cells will know, okay, that I am not going to use it for glycolysis or anything, that is destined to be stored. So once you have UDP glucose, this is your starting point. So here we need to add more glucose units to extend the chain. How would that happen? How could that happen? Anyone remembers tyrosine side chain? OH and benzene. So that's your tyrosine, and this tyrosine is coming from glycogenine. So this is our glycogenine, which has the tyrosine. It will do something. It will attach. So can you attach this to that molecule, UDP glucose? We just discussed here that if you have more phosphates, that's a leaving group, right? Good leaving group. So this one has 2 phosphates, so that can be a leaving group. Attaching this would also activate that carbon, which is needed for chain growth. Okay, so here what we have is, so that's your glycogenine plus UDP glucose. If you want to make glycogen, which part of the molecule does that oxygen have to attack? What is the linkage, the linear linkage? Alpha 1, 4. So where is 1 here? That's 4. So let's attack it this way. We already have our starting point. So we have only attached 1 sugar unit, right? We need to do about 7 more times to make the glucose 8 primer. So here then, okay, what did we lose here? We lost something here.
UDP.
Just UDP. We see 1, 4, right? So we are starting to see the building block. So now only 1 glucose unit is here. So how can the next glucose unit come? Could we do the same way? We made UDP glucose for a reason, right? So that's your starting. That is what is going to be added every time. So this is like your monomer unit, which will be attached all the time, right, throughout the synthesis process. So we have another UDP glucose. How are we going to make the bond? So once we have that, then what are we going to make? Glycogenine is right here. So this is our tyrosine. Glycogenine attached to tyrosine, first glucose unit, second glucose unit. What will happen next?
Same thing.
Same thing. So this will attack here for another molecule of UDP glucose. And So this way you will produce about 8 glucose units. And so this will be your primer. Now remember, when we did PCR, we used a primer which was a nucleotide. In this case, primer is a small oligomer of sugar, 8 units of sugar. What was the enzyme in PCR? DNA polymerase. So if you have a primer, it means there must be another enzyme which is the actual enzyme which is making that long chain of glycogen, right? So in this case, that enzyme is glycogen synthase. So glycogen synthase will keep adding glucose into this primer. So glycogenin has two functions. One is to first use the tyrosine to attach the first UDP glucose, then extend up to 8 units of glucose. So it doesn't stop after attaching only one, but it will attach the rest of the other 7 to go up to the 8 units of glucose. So we have made the primer 8 glucose unit, and glycogen synthase is going to keep adding. It will take this plus UDP glucose. So UDP glucose is always your starting point. Without that, you cannot extend glycogen chain. And now we will keep adding UDP glucose, and this is going to be done by glycogen synthase. Glycogen synthase will only make the linear part, it will not make the branching parts. So we need another enzyme to make the branches. So glycogen synthase will only make the linear or alpha 1,4 linkages. It does not have the ability to make the branches. Can you guess which enzyme could do it? Very close. Glycogen branching enzyme, that's the name. All of these are alpha 1,4, that is your alpha 1,6. And all this glycogen branching enzyme did was to grab residues from this end and put them here to the C6. Here is C6, and this is your alpha 1,6 linkage. So we made one branch. Again, we see we have this non-reducing ends. So again, glycogen synthase will keep adding until a certain length is reached. Then again, branching enzyme will come and make the branch. This is how the process will repeat. Okay, so this is how we make glycogen. We make glycogen under fed condition. We have glucose, and glycogen will only be made after the need for the liver and tissue, the muscle and the brain have been met. So there is excess glucose that has to be stored. So the summary, the storage will only happen when the body is not using glucose, right? And to store glucose, we have to use G1P first. Then G1P has to be attached to this UTP to make UDP glucose. Then UDP glucose will be attached to glycogenin to make the primer. Glycogen synthase will make the linear part. Then branching enzyme will put the branches on. Again, glycogen synthase will keep going. Again, branching enzyme will make the branches. This is how it will continue until you reach approximate mass of glycogen, which is what about 55,000 glucose unit total. 55,000 units of glucose total in glycogen molecule, approximately.